So I'm going to talk about some guard cell transcriptomics data, as well as a resource that my lab runs called the PAR Bioanalytic Resource for Plant Biology. So drought is not expected to decrease in the coming years. In fact, we're expecting an increased incident of drought worldwide. So it's important to understand a plant's response to the level of guard cells. So I thought it would be useful to go back to some PhD work that I did in 1997, looking at the guard cells of droughted plants, potato plants. I was in Germany. Germans love their potatoes. So we were kind of interested in how the organic and inorganic osmotica work to bring about osmotic potential in guard cells. And we droughted potato plants, and you can see here that after about 2 days, we see no big change in the leaf water potential, but quite a dramatic decrease in assimilation and transpiration. And then an interesting thing about this experiment was a re-watering, and you see a kind of a slow recovery of those parameters, even though the leaf water potential hasn't really changed in the top leaves that we were looking at. So I developed a method at the time to isolate RNA from guard cells. It involved using a blender and just blending up the leaves and collecting the epidermal fragments. The only things that survive that treatment are the guard cells. You can see this with a RNA-specific stain, cyto12. And then I did a bunch of northerns. Who remembers what northerns are? Yeah? All right, yeah.
Old folks.
So here we go. So we see in strong downregulation of potassium channel, some change in the proton ATPase, some changes in genes involved in starch metabolism, so starch synthase. And so we came up with a model, what was going on in those droughted guard cells of the potato plants. And fast forward a couple of decades, now with transcriptomic experiments, RNA-seq, we can do thousands of northerns in one run effectively. And I've just highlighted the genes that we were looking at in our experiment. And I'm here today to tell you about discovering hopefully some more meaningful patterns from the data. These weren't, this is just a joke, right? So I'll talk about, as I mentioned, two aspects of my lab, the bar, and then also this guard cell drought transcriptomics experiment that we did in terms of three aspects: gene expression data, protein-protein interaction data, and protein tertiary structure data. So as I mentioned, my lab runs something called the Bioanalytic Resource Plant Biology, the bar at utoronto.ca. And we've got lots of gene expression measurements, subcellular localizations from online database run by Harvey Miller, the University of Western Australia, lots of predicted and experimentally determined protein-protein interactions, protein structures, lots of non-synonymous SNPs from the 1001 Genomes Project, and some other data too. The bar in 2023 became designated a global core biodata resource, one of 52 worldwide. We were quite excited about that. It doesn't actually give us any money, but I get to show you that sticker. So we're going to turn now to using gene expression data for hypothesis generation, looking at the bar. So as you all know, gene expression data can guide wet lab experiments. So this is a case of 3 genes in Arabidopsis DHS 1, 2, and 3 that encode 3-deoxy-D-arabinoheptalosinate 7-phosphate synthase, which catalyzes the first step in the shikimate pathway for corsmate synthase, important for a lot of plant secondary compounds. My colleague is very interested in this pathway, and he ordered the knockouts of these 3 genes. And when you look at the mutant plants under normal conditions, you don't see any phenotype, right? So what do? You can turn to the gene expression data, and you actually see an increase in expression of those 3 isoforms under UV light. This data set generated by Detlef Weigel's group at the time And lo and behold, when you expose those mutant plants to UVB light, they don't do as well. So there's definitely something going on for the plant's response to UVB light with these three enzymes, probably producing protective compounds. So we developed a way for exploring transcriptomic data called the EFP browser, the electronic fluorescent pictograph browser, in 2007. And this was kind of the original view, again, with data from Detlef Weigel's group, Schmidt et al., and then also from the Nambara et al. For the dry and imbibed seed samples. And there are a number of features. You can choose different views that we've put together over the years. You can choose to view in absolute mode, just the absolute expression level, or relative to appropriate control. You can also click on those tissues to get more information about the samples. And there's also a little known feature, which is the expression level distribution graph, which tells you how strongly this particular gene, so DHS3 we're looking at, is expressed relative to all of the other genes in the data set. So it's expressed quite strongly actually in the stems. That's what the color scale tells you. So the red color denotes strong expression in stems and in sepals, what are those, petals and stamens there. And you can click on the magnifying glass to drill down to some cell type specific data sets that have been generated over the years that we've curated. And the root data set from Ken Birnbaum and the late Phil Benfey's lab. Light series, we see some oscillation over the course of the day. We also see some increased expression in response to pathogens. That's the UVB series there. So let's go back here. There we see the increase in expression in response to UVB. And then we also see strong expression in the seed coat. So what the role of DHS3 is in Arabidopsis seed coat, who knows? No one's looked at it, but it seems to be really strongly expressed there. So that would be a candidate location to look at for some change in the mutant. Chemical series, natural variation. And we've also got lots of RNA-seq data in the EFP browser. This is from Klepikova et al. And then we've also got some single-cell RNA-seq data this is from John Schiefelbein's lab at University of Michigan. We've also got now a small farm of EFP browsers, so rice, soybean, and other species, potato, grape, Camelina sativa, Viscometrella patens, wheat. With wheat, we've actually worked with Cristobal Uwawe's group, John Innes, to link up the homologs. So you can easily switch between the homologs and the expression pattern in the homologs. And you can see that there are expression differences with these homologs in wheat, which is kind of cool to see. We reimagined the EFP browser in the mid 2010s, so in about 2015. And we wanted to have a zoomable user interface where you could migrate between different scales of data. So at the centimeter scale, sort of organ level expression, down to the millimeter scale and so on. And we improved the graphics for the developmental atlas views. These are SVG graphics now. It's very easy to get the information about the experiment. The graphics are available under a BYCC license. You can just include those in papers without having to contact me for permission. So that's the gene expression data part of the bar. In terms of guard cells, we wanted to revisit, or I wanted to revisit what we saw in potatoes. And in Arabidopsis, and we wanted to do a physiologically relevant drought, and we wanted to use intact to isolate guard cell RNA because the epidermal fragment method doesn't work so well in Arabidopsis. And we didn't want to use fluorescence activated cell sorting or laser capture microdissection, quite complicated. So Intact introduces a tag into the nuclei. This is not a physiologically relevant drought where you take the plants out of a hydroponic culture and dry them with a hairdryer, for instance. We actually performed a drought experiment mirroring Ulrika Bechtol's experiment from Durham, where they let the plants dry out over a course of about 2 weeks. And we took samples at about 60%, 40%, and 20% soil water content. And then we used this Intact system, which introduces a biotin ligase recognition particle into the nuclei of specific cell types. This is driven by a specific promoter. And you can see the 2 lines that we use. This is the guard cell specific promoter driven by CAT1. There's only expression in guard cells. And here's the 35S control, which is expressed everywhere, including in mesophyll cells and so on. We started to try and isolate RNA after making transgenic lines. And this is, if we just grind up leaves, we can totally get RNA. But when we use the standard Intact protocol, this is what our RNA looked like. The graduate student was not happy, but Anna Van Waring spent about 2 years optimizing a fixation protocol, PFA, and she was very excited when she finally was able to get really nice RNA using that fixation protocol. So we generated 48 guard cell transcriptomes, triplicate samples at 20% soil water content. And then there's a 24-hour rewatering time point using the guard cell and 35S Intact lines. And we had age-matched well-watered controls. So this is important because there are changes over developmental time, the 2 weeks that we were looking at things. We submitted these to our Genome Center for sequencing and then mapped the reads. This was a guard cell specific gene, one of the first views that I looked at. So excited to see that it's only expressed in the guard cell samples. These darker green samples here, there's no expression. And we get lots of data and lots of differentially expressed genes. So there are, and here's the CAT1 gene that we were using to drive the expression of our guard cell line. 4,837 guard cell drought DEGs, 6,651 whole shoot drought differentially expressed genes, and then in total 7,000 drought differentially expressed genes in any tissue. So lots of candidates. When we look at gene ontology enrichment analysis, here are our up differentially expressed genes. These are the down differentially expressed genes in guard cells and in the whole leaf samples. We can see that there are responses in guard cells to, these are stress categories here that you don't see in the whole leaf sample. Nicely, we also see changes in carbohydrate metabolic processes, which we actually saw in potato plants, if you remember the northern blots from earlier. And of course, the question is, what's going on in guard cells at the early drought time points, right? So, and we'll come back to that to see what we found out. So now we're turning to protein-protein interaction data in the bar. We generated the first interactome in 2007, and we've curated many protein-protein interaction data sets over the years, published in the literature, roughly 100,000 at this point. This predicted interactome, first predicted interactome, was based on interacting orthologs from 4 other species, now 7 other species, where we just did like a match replace of the Arabidopsis genes with these 4 organism genes to come up with the predicted interactome, which we validated by pairwise correlation and then also subcellular localization. So for proteins to interact, they should be at least some of the time in the same compartment, right? So we also generated a structure-based interactome in 2019. And here we took around 30,000 predicted protein structures. This is pre-AlphaFold days, so actually way back in 2011. And we used FIRE2 to generate those structures in collaboration with Lawrence Kelly and Michael Sternberg at Imperial College London. And we took a subset of those structures, roughly 1,300, that had good coverage and good identity to the templates. And we did docking on about 1 million protein pairs. And we did this using a program called HEX, and we identified a list of about 9,000 interactions that seemed to have good scores for being able to interact with one another. And we did, again, we validated this by gene ontology enrichment analysis in terms of the subcellular localization, and we tested a subset of these by these two hybrid to see if they were in fact bona fide interactors. This is just how HEX works. You take protein 1, protein 2, and you test all possible, not all possible, but many possible configurations in 5-degree increments. As you turn protein 1, test all docking, test the docking, the free energy, and then you ask, is it good or is it isn't good? You turn the protein, the second protein again, you continue through many different configurations to come up with the overall docking score. Right, so we've put those interactions into our revamped interactions viewer. Arabidopsis is not very imaginatively named, Arabidopsis Interactions Viewer 2. And you can see not only protein-protein interactions, green edges are experimentally determined, dashed lines are predicted interactions, but you can also see around 2.8 million protein DNA interactions generated by Joe Ecker's lab using DAP-seq method developed by Carol Huang and Ronan O'Malley. And nicely, you can see if a given transcription factor binds to just one target or to multiple targets, and if there's common binding of multiple transcription factors using this little grid here in the protein DNA interaction section. We continue to curate protein DNA and protein-protein interactions. We're going to add double DAP-seq data soon. Got some complex data, some kinase interaction data, BIN2 kinase interaction data generated by TurboID. And there's some kind of cool features of the AIV2 in the sense that you can change the layout from just sort of a general the kind of blob view that people are familiar with to more of a stacked view from the outside of the cell to the inside of the cell to kind of generate like almost like signaling pathways. And you can also overlay expression data from, in this case, the first node of Arabidopsis plants to help define subnetworks within these protein-protein interaction networks. And that might be useful to see if there are different subnetworks acting in different cell types. Right, so how did we use the protein-protein interaction data for our particular guard cell gene expression data set? We used it to filter down that list of whatever it was, 5,000 genes to come up with, first of all, we looked at 212 early responding genes, and then we prioritized that list by looking at the connectivity of those particular genes to the genes in our protein-protein interaction network available through the Arabidopsis Interactions Viewer 2. So we took kind of the hub genes, thinking that those hub genes are probably more important for function than ones that are just interacting with a few genes. And then we also cherry-picked a few transcription factors here that were differentially expressed at early time points, as well as some other signaling proteins. And we'll come back to these and see what happened with those in a bit. But now we'll turn to protein tertiary structure in the bar. So again, as I mentioned with ePlant, we wanted to be able to navigate from the kilometer scale, sort of natural variation, all the way down to the nanometer scale, which is protein structure. And this is just a snapshot of the ePlant interface. And we actually have worked with Merrick, who's in the audience, to incorporate not only a curator summary from TARE, but also plant connectome, a summary of the plant connectome summary that we generated with Llama 3. And if you click on that link there, you can actually go to plant connectome. Here at the kilometer scale, we see some variation. This is ABI5 actually, you see some expression differences. We can actually overlay climate data onto the map. And so the idea is that visually, if you see some kind of correlation between expression patterns and say rainfall or temperature, you could then go and investigate that using GWAS approaches, obviously. At the centimeter scale, we can see that this gene ABI5 is strongly expressed in dry seed, also in guard cells. At the millimeter scale, at the sub-millimeter scale, we can look at protein localization. It's a transcription factor. SUBA tells us that it's quite strongly localized in the nucleus. At the micrometer scale, we can see where the gene is localized on which arm of which chromosome. And also at the micrometer scale, we can see the protein-protein interactors of ABI5, lots of them. And then sort of last, we have at the nanometer scale, protein structures. And then sort of one of the concepts of ePlant is integrating across scales. So we've actually brought in the non-synonymous SNP data from the 1001 Genomes Project that you can explore in the context of the protein structure. So we have not only our Fiber2 predicted structures, but now we've also loaded in AlphaFold 2 structures. And you can see that there's a non-synonymous SNP here, histidine to an aspartate, that's actually in the DNA binding domain. So this DNA binding domain comes from the conserved domain database. So again, integrating from another database. And also at the nanometer scale, we can drill down to the DNA sequence. And this view is provided by TARE's JBarrows. Right. So the architecture of ePlant really allows us to separate databasing and visualization. We use web services to provide information to the different viewing modules. And we're working on making it even easier to add new modules to ePlant. So we're rebuilding it in React and so on. So that's an ongoing effort. Right. So how can we use protein structure in the context of identifying small molecule targets? This is something that we're working on where we're interested in taking our protein structures. So here's a BRI1 structure. These are actually experimentally determined structures, and TDR, another receptor. And these are their respective small molecule binding targets. And when we do docking with hex, we actually see that BRI1, which binds brisinolide, actually it can find the brisinolide binding site quite nicely. TDIF is a small peptide that TDR binds, and we see that that finds its location for binding right there, but not in the BRI1. And conversely, in that case there, BRI1 does not bind TDIF that well. So red denotes that's where it's found most often, 500 tests with hex. So we've built many ePlants, 15 ePlants so far, with a few more to come soon. Some of them have structuromes. There's barley ePlant, tomato ePlant. This has a structurome associated with it, with some cell type specific data from Siobhan Brady's lab. And in the case of the guard cells, what we're actually interested in is the ABA signaling pathway in the lab, because that's also important for drought response. And this is the canonical ABA signaling pathway published in 2009. And there's interaction between when ABA is present between PIR and PPTC, and we get a release of the repression on SNRT2, and then that activates downstream factors for an ABA response. And so we're focusing on SNRT 2.6 and ABI5. And the idea here, this is work in progress by another student in the lab, Wang, to look at the non-synonymous SNPs in ABI5 and SNRP 2.6 in the context of their protein structures and how they interact with one another. So we're using AlphaFold to determine the protein-protein interactions and where the interfaces are between the proteins, and then looking to see if there are non-synonymous SNPs in the interface, and then whether or not those non-synonymous SNPs affect protein binding. So that, as I mentioned, docking, and then we're looking at these non-synonymous SNPs to see if we can see a difference in signaling outputs in different ecotypes of Arabidopsis. And we'll test that with yeast 2 hybrid. This is just a preliminary experiment where we're taking known mutations of SNRP 2.6 and 5 and looking at the strength of the interaction using a yeast 2 hybrid based output. The next thing that Yifan is working on is to look at these non-synonymous SNPs in the context of this interaction here. So back to our guard cell story. I told you we sort of winnowed down our list of 212 differentially expressed genes to a set of 80 genes using various data sets that we had available to us. And then we thought, well, if we look at the knockouts of those genes, do they actually have a drought phenotype, a drought response phenotype? And our control for this was OST1, open stomata 1. So normally in Colombia at 40% soil water content, we see the closing of stomata, the leaf temperature increases, whereas open stomata 1 mutant has open stomata, like the name suggests, and the plants are cooler because of that, because of transpiration. And then with some of our candidates, at least 10 of them, we actually saw cooler temperatures. And we're in the process of actually further investigating those, looking at stomatal counts. The stomatal density, at least in 4 of these, is not different. And those are the ones we're following up in terms of our investigations. And there's also an interesting alternative splicing story going on in the data. So the nice thing about RNA-seq data is that you can actually look at alternative splicing. So for these 2 genes, SAFE1 and XTH31, we see changes in splice isoforms in response to drought. And again, there's some phenotype. When you knock out those genes, we actually see differences in terms of the drought responses. So we're following up on that actually in tomato. So we're looking at 40% soil water content, droughted tomato plants versus the water control. And here we're using an epidermal fragment approach, and we can actually separate out those RNA-seq samples, at least using PCA into the 2 treatments, which is good. And we've done this with long read sequencing, so we should be able to really look at our alternative splicing events in tomato quite easily. So, time. Okay, so now I'll talk about integrating co-expression, protein-protein interaction, and other data. It's all a bit overwhelming. First of all, we can explore orthologs in terms of both their of a given gene using both sequence similarity as well as expression pattern similarity. We've tried to rank our genes based on expression pattern similarity, how those genes are responding in equivalent tissues in other species. And you can read about how we did that in this paper. And this was refactored by an undergraduate student for use in ePlant. It's got link outs to various other resources. But we've developed something called Gaia, which was intended to be like a ChatGPT for plant biology before ChatGPT existed. So the underlying, right now, the underlying NLP engine is not that good, but the idea was that you could say, hey, tell me about ABI3, and Gaia does provide you some information about the annotations, some figures, and so on. We actually have the plant connectome output in this now. I'm not showing it in this figure. It'll tell you where the gene is most strongly expressed. We've also gone to the literature, and in about 65,000 Arabidopsis publications, we extracted figures, and we used machine learning to identify the figures that are genetic models. And in those genetic models, we've actually used optical character recognition to identify gene names in those figures. So, and you can ask for that, for your gene of interest to be highlighted in a figure. You can also ask the question, does my gene of interest appear together with another gene in those figures? So you can kind of really quite quickly identify figures that have both of your genes of interest. Gaia is now the main entry point to the bar. So in this simple search box, you can enter a sequence, you can enter a gene name. If you enter a sequence, we actually have a BLAST server that will then link your hits to the respective ePlant or EFP browser for the matches to your gene of interest. Right. So that's kind of the overview of what I wanted to talk about. We're sort of at the stage now in biology, I feel, where we're trying to figure out the wiring of plants. We could consider the plant cell as a city with the various actors, who's living where, doing what their job is, how they get from A to B. Or we could consider it as a circuit. This is a Colpitts oscillator here that I put together. It's an online tool. We've got some capacitors here, transistor, resistor. And that's all nice to document all that stuff. But ultimately, what we're interested in is when we turn on our phone to be able to listen to some music, right? And kind of that dynamic sort of response is, I think, what we're interested in as plant biologists. And hopefully with all these data sets that are being generated, we can kind of start to understand the song of the cell. This is a great book if you want a good sort of scientific read. The last chapter talks about how we're now moving into this era of biology, of like systems biology, and understanding how a cell responds at the level of systems instead of just its different components. So how can we lift the fog on what's going on in the cell? Well, I think some tools that are being developed are going to help. So SCGPT is a nice tool, DNA BERT. These are all kind of AI approaches to help decode the genome. And plant connectome will help too, hopefully. And we think that initiatives like the Plant Cell Atlas will also help provide data for these tools to help understand biological processes better in plants. And we think that Arabidopsis is actually the perfect computable plant, at least potentially. And we've just released a paper called Arabidopsis Research in 2030, translating the computable plant, where we make some suggestions to the community in terms of using Arabidopsis as a reference, ensure if maybe the gene isn't present in Arabidopsis, but you can also develop techniques in Arabidopsis very rapidly and then transfer those techniques to other plants and that kind of thing. So that's perhaps some reading if you wish. And I'm currently involved in a project that's related to the Plant Cell Atlas called C-SPIRIT, the Center for Sustainable Plant Innovation and Resilience through international teamwork. We got a grant from NSERC, but there are 4 other partners, the NSF in the US, BBSRC in the UK, and Japan and Korea, with the goal of identifying small molecules that can help with plant resilience. And my part in this is to actually set up a database of single-cell RNA-seq, as a sub-aim of AIM-3. So that should be coming in soon, maybe in a year or 2, who knows, but hopefully soon. All right, and with that, I'll finish up. These are many people who have helped with the bar over the years. Again, you can visit us there. We're available on Blue Sky for comments. And I thank you for your attention, and we'll take any questions.